Hello everyone, so here we are to complete our story on the metabolism of lipids. So today we're gonna conclude our chapter with the story on metabolism of triacylglycerols, glycerophospholipids and sphingophospholipids. So today's topic is dedicated to complex lipids. We're going to start with the degradation of those molecules. We're going to start with catabolism. And of course, the most famous complex lipid is 3-acylglycerol. So we're going to start with the catabolism of 3-acylglycerols. Because we have mentioned 3-acylglycerols numerous times in previous topics and in previous chapters, and we know so far very well that 3-acylglycerols are actually the major sources of fatty acids. So by their degradation, our cells are supplied by sufficient amounts of fatty acids, which are the major sources, major energy sources for our cells during the process of beta oxidation and subsequent Krebs cycle. In the degradation of 3-acylglycerols, numerous enzymes are involved, and most of those enzymes are very well known to you from previous topics. In the chapter on digestion of lipids and absorption of lipids, we have mentioned a couple of digestive enzymes which hydrolyze 3-acylglycerols. Let me remind you. Lingual and gastric lipase are enzymes located in the stomach. The first one synthesized in lingual glands, the other one synthesized by chief cells of the stomach. So the result of the enzymatic activity of both enzymes is hydrolysis of 3-acylglycerols to monoacylglycerols and fatty acids, following the mechanism that we have already explained in previous chapters. Then, following the digestion of lipids, we have the activity of one other digestive enzyme, which is pancreatic lipase, active in duodenum, also responsible for degradation of 3-acylglycerols to 2-monoacylglycerols and fatty acids. Plus, to remind you that to the certain extent, minor extent, up to 20%, degradation of 3-acylglycerols in our gastrointestinal tract may yield the production of fatty acids and glycerol itself. Then in our last video, I have mentioned several times very important activity of lipoprotein lipase on the cells of capillary endothelium, which was responsible for degradation of 3-acylglycerols from lipoprotein particles, if you remember, from kilomicrons, from VLDL particles, with the role of hydrolyzing 3-acylglycerols to glycerol and fatty acid. If you remember, this enzyme was activated by apoC2 apolipoprotein. In the chapter on lipoprotein metabolism, we also mentioned the activity of hepatic 3-glycerate lipase from liver sinusoids, which was responsible also for hydrolysis of TAGs to glycerol and fatty acid, but this time it was responsible for the hydrolysis of IDL particles, if you remember, to LDL particle. But this time I'm going to mention one more enzyme involved in the catabolism of TAGs, and that is hormone-sensitive lipase from adipose tissue, which is responsible again for hydrolysis of TAGs to glycerol and fatty acids. But this enzyme is, unlike other that we have mentioned, its activity is regulated by hormones, predominantly glucagon and epinephrine, and insulin as their antagonist. So I'm gonna remind you just in short about the mechanism of the regulation of the activity of hormone sensitive lipase, which is absolutely identical with the one that we have discussed for regulation of glycogen synthase or glycogen phosphorylase. So uh, activated hormones glucagon and epinephrine actually bind to specific receptors on cell membrane, and if you remember, this binding to specific membranes, specific receptors of cell membranes, those were glucagon receptors, those spiral receptors, and those beta and alpha receptors for epinephrine actually initiate the transfer of signal through G protein to activate adenylate cyclase, which is responsible for the synthesis of cyclic AMP. Cyclic AMP then binds to protein kinase, thus activating this enzyme, and activated protein kinase is responsible for phosphorylation of hormone-sensitive lipase, which is active when phosphorylated. So, upon the activation of HSL, TAG hydrolysis starts, so glucagon and epinephrine, they promote the activity of HCl, thus promoting lipolysis. Insulin inhibits the activity of HCl, thus promoting lipogenesis. Now here I'm gonna also remind you about certain differences in different textbooks regarding the exact description on HCl activity. So in certain number of textbooks you will find that HCl is responsible for the complete hydrolysis of TAG to glycerol and fatty acids. In some other sources you will find that HCl, it's only the first out of 3 specific adipolytic lipases responsible for the hydrolysis of triacylglycerols to 2-acylglycerols and fatty acids. Then the second enzyme, deacylglycerol lipase, hydrolyzes deacylglycerols to monoacylglycerols and fatty acid, and finally monoacylglycerol lipase catalyze the hydrolysis of monoacylglycerols to glycerol and fatty acids. So have in mind both versions and be careful when reading your test questions, and it's not going to be a big deal for the oral part of the exam because it's going to be based on the conversation with your professor. So anyway, let's proceed to the catabolism of glycerophospholipids. We have already mentioned this, but it's always worth reminding ourselves and refreshing our knowledge. So glycerophospholipids are major components of both cell membranes and organelle membranes. And also they have an important role in metabolism regulation because they are involved in the synthesis of important signal molecules. Catabolism of glycerophospholipids is completed by the action of different phospholipases. So far we have described the activity and existence of 4 different phospholipases, A1, A2, C, and D. So phospholipase A and 1 actually hydrolyzes ester bonds at C1 or C2 carbon atom of glycerol, thus forming free fatty acids and lysophospholipid molecules. It is important to mention again that phospholipase A2 actually removes predominantly arachidonic acid, which is usually found at position number 2 of certain glycerophospholipids, and I'm going to get back to this a few minutes later. And arachidonic acid is the precursor for the biosynthesis of prostaglandins. So if this phospholipase A2 is inhibited in any way by natural glucocorticoids or by synthetic corticosteroids, then the release of arachidonic acid is going to be inhibited. Thus, the overall biosynthetic pathway of prostaglandin is going to be inhibited, thus inhibiting the immune response of the body. Phospholipase C removes the polar part of the molecule, phosphate group and alcoholic group, thus releasing the acylglycerol from the molecule of glycerophospholipid. And phospholipase D, the last one, as you can see from the figure, it removes the alcohol group, yielding the formation of phosphatidic acid. Catabolism of sphingophospholipid is completed in separate ways from glycerophospholipids and triacylglycerols. So let me remind you that the major group of these molecules are glycosphingophospholipids, and because in their polar head they also contain certain carbohydrate remnants or carbohydrate units attached to that polar head. So a bunch of different lysosomal hydrolytic enzymes are responsible for degradation of sphingophospholipids. At the first stage of catabolism, enzymes are enzymes which remove carbohydrate units one by one are involved in the degradation, thus yielding in the final stage yielding the formation of ceramide, which is the basic structure of sphingophospholipids. In the second stage, this is some kind of divert to this degradative processes. Sphingomyelin is degraded also to ceramide by the removal of phosphocholine, and you will see the opposite pathway in the biosynthetic pathways later on. And finally, in the stage 3, ceramide is further degraded to give sphingosine, the alcoholic analog to glycerol, and fatty acid. So, inherited deficiency of any of these enzymes from the catabolic pathways of sphingophospholipids yields the formation and development of so-called sphingolipidosis. And as a result of this, the accumulation of certain sphingolipid occurs, which yields mental retardation, and often, and this disorder is often lethal. Speaking of the biosynthesis of those complex lipids, we can start with triacylglycerols because they are really predominant complex lipids in our organism. So biosynthesis actually requires activation of both fatty acid and glycerol. So fatty acid is activated in the way that it is very well known to us so far. So fatty acid undergoes reaction with ATP, so we get fatty acyl adenylate or fatty acyl AMP, and then we have the transfer of fatty acyl or acyl unit to coenzyme A, so we get fatty acyl coenzyme A, and AMP is released. Speaking of the activation of glycerol, glycerol is supplied from glycolysis or from adipocytes after the catabolism or degradation of triacylglycerols, and we mentioned that glycerol cannot be activated in adipocytes in the way it's activated in the liver. So actually we have two pathways of glycerol activation. So the first pathway, as you can see from the figure, occur only in the liver by the enzyme glycerol kinase. So the reaction is direct phosphorylation at the position number 3 to get glycerol 3-phosphate. The second activation pathway occurs in both liver and adipose tissue. So it means the conversion or reduction of dihydroxyacetone phosphate to glycerol 3-phosphate. DHAP is obtained from glycolytic pathway, and the enzyme is glycerol 3-phosphate dehydrogenase, which requires NADH as a donor of reducing equivalents this time. And this is the reaction that we have mentioned before in gluconeogenesis gluconeogenic pathways, if you remember. So when both components are activated, so when fatty acid is activated and when glycerol is activated, they can be now subjected to the biosynthesis of triacylglycerols. But before we move on, let me remind you that there are certain differences between the TAG synthesis in enterocytes and other cells. So in enterocytes, if you remember, we had not exactly biosynthesis, but resynthesis of dietary TAGs. So the reaction went in the way that two monoacylglycerols react with two acyl coenzyme A's to get TAG and coenzyme A. But in hepatocytes and adipocytes, glycerol 3-phosphate undergoes the first condensation with the first fatty acyl coenzyme A, yielding the formation of lysophosphatidec acid. The reaction is catalyzed by one acyl transferase, but we have another step upcoming another step. So lysophosphatidec acid reacts with the second acyl coenzyme A, producing phosphatidic acid and coenzyme A, like you see from the figure, and the same enzyme acyl transferase catalyzed this reaction. The next reaction is hydrolysis of phosphatidic acid to deacylglycerol, catalyzed by phosphatase. And finally, the last step is the reaction with the third fatty acyl coenzyme A, thus yielding the formation of 3-acylglycerol and coenzyme A. 3-acylglycerol has different fates depending on the location where it's synthesized. So if TAG is synthesized in the liver, it's going to be packed in the VLDL lipoprotein particles and secreted to blood. But if TAG is synthesized in adipose tissue, it's going to be accumulated and deposited in so-called adipose store, where it can be accumulated, waiting for the situations in which these TAGs are going to be hydrolyzed and catabolized again to give fatty acids for energy supplies. Speaking about the biosynthesis of glycerophospholipids, these biosynthetic processes occur in all cells but mature enterocytes. Biosynthesis occurs in cytosol, in endoplasmic reticulum. Glycerophospholipids are, as we've mentioned, major components of cell membranes, but they are also important for the transport of lipoprotein particles, because if you remember, they form that hydrophilic surface of lipoprotein particle, thus enabling its integrity, and it's involved in the maintenance of its integrity and increasing the solubility of insoluble lipid molecules. On C1 carbon atom of glycerol, mostly saturated fatty acids are found, and on C2, predominantly unsaturated fatty acids are found. Precursors for the biosynthesis of glycerophospholipids are phosphatidic acid or 1,2-deacylglycerol, depending on the pathway, and plus of course an adequate alcohol, depending on the final glycerophospholipid to be synthesized. There are actually two mechanisms. The first one is by activated 1,2-deacylglycerol, and the other one is by activated alcoholic group. So you can, as you can see from the slide, we can activate head group from alcoholic group. So phosphorylated head group undergoes reaction with CTP to CDP head group, which then reacts with the acylglycerols, thus producing glycerophospholipid. This pathway number 1, is reserved for the biosynthesis of phosphatidylcholine, phosphatidylethanolamine, and phosphatidylserine. The other pathway actually means the activation of phosphatidic acid to CDP deacylglycerol, and subsequent reaction with head group of alcoholic compound, yielding the formation of specific glycerophospholipids. And this pathway occurs during the synthesis of phosphatidylinositol, phosphatidylglycerol, and its dimer cardiolipin. So the key point of the biosynthesis of glycerophospholipids is the activation or binding to CDP in order to get CDP diacylglycerol or CDP alcohol. So let's go one by one. This figure represents the biosynthesis of phosphatidylethanol and phosphatidylcholine, or if you want to use the trivial name, the synthesis of caphaline or synthesis of lecithin. So if you can see from this figure, diacylglycerol undergoes reaction with activated ethanolamine or activated choline to get phosphatidylethanol or phosphatidylcholine. I will get back to these details in the next slide, but let me also introduce you to another way to synthesize phosphatidylcholine, which is actually from phosphatidylserine upon decarboxylation reaction yielding the formation of ethanolamine, and then from ethanolamine 3 subsequent methylations occur with the S-adenosylmethionine as a donor of methyl groups in order to synthesize phosphatidylcholine. But let's go through this figure step by step. So, the biosynthesis of phosphatidylethanol and phosphatidylcholine can be achieved in upcoming way. So, choline and ethanolamine are obtained predominantly from diet or from existing phospholipids. So, in a way, there is a unique reutilization of choline, and it's so important because for choline synthesis we require 3 methyl groups, and which are provided by S-adenosylmethionine, which is an active form of essential amino acid methionine, which has to be found in our diet in sufficient amounts. So the first step of the biosynthesis of glycerophospholipids is phosphorylation of OH group of polar head with ATP. Reaction is catalyzed by ethanolamine or choline kinase depending on the final product. Step number 2 is reaction of that phosphate group from phosphoethanolamine or phosphocholine with CTP, yielding the formation of CDP ethanolamine or CDP choline. Reaction is catalyzed by phosphoethanolamine or phosphocholine cytadilyl transferase. Third step of the reaction is now reaction of those activated alcoholic groups with 1,2-diacylglycerols, yielding the formation of an adequate glycerophospholipid and release of CMP from the system. Reaction is catalyzed by CDP ethanolamine or CDP choline, 1,2-diacylglycerol, phosphoethanolamine, phosphocholine transferase. There is, as I've mentioned previously, and I'm going to get back to the figure, there is an alternative pathway for the biosynthesis of phosphatidylcholine or lecithin, and that is decarboxylation of phosphatidylserine yielding the formation of phosphatidylethanolamine, followed by the 3 subsequent methylations by S-adenosylmethionine as a donor of methyl groups, yielding the formation of phosphatidylcholine. This is the major pathway for this biosynthesis of dipalmitoyl phosphatidylcholine, which is molecule found in pneumocytes type 2 of lungs. This is the major lipid component of lung surfactant, which is nothing else but a unique extracellular liquid layer lining the surface of alveolar sac. So this molecule actually, and the role of surfactant is to reduce surface tension, prevents alveoli to collapse with expiration. In order to explain that, I'm gonna show you just in a very simple way. So upon expiration, the alveolar sac tends to remove the air, and in order to in the presence of surfactant, it's gonna decrease just too little, let's say half of the way or so. So not much energy is gonna be needed for its relaxation again in order to catch up the air with inhalation. But without the surfactant, it's gonna collapse totally. So a lot of energy is gonna be required for it to be fulfilled with air. So in the absence, or in the case of insufficient production of surfactant, respiratory distress syndrome occurs of premature infants dominantly, and it is believed that respiratory distress syndrome is responsible for approximately 15% of lethal cases in newborns. Of course, this respiratory distress syndrome can occur in adults as well, in situations where pneumocytes are destroyed, usually upon the application of immunosuppressants and chemotherapy. So patients undergoing those therapy of chronic diseases are a really vulnerable group for all diseases and viruses attacking lungs, thus this actual coronavirus as well. Here on the slide you have a structure of this molecule, dipalmitoyl phosphatidylcholine, but I think it's very well known to you, having in mind the structures of the components of this molecule. Speaking about the biosynthesis of phosphatidylinositol and phosphatidylglycerol and cardiolipin, we mentioned that it occurs in a slightly different way from the previous. So phosphatidylinositol is important in cell signaling, and I'm going to remind you of the phosphoinositol pathway that we have explained previously when we discussed about the regulation of carbohydrate and lipid metabolism. It's an unusual phospholipid because at position C1 we have stearic acid instead of palmitic, and at C2 we have arachidonic acid. So phosphatidylinositol is a unique reservoir of arachidonic acid for the biosynthesis of prostaglandins. Phosphatidylglycerol is a precursor for the synthesis of its dimer cardiolipin, which, if you remember, we mentioned it, it's one of the major glycerophospholipids in mitochondrial membranes. So the figure represents the biosynthetic pathway of those two molecules. So phosphatidic acid this time is activated by CTP, forming CDP-deacylglycerol. So the CDP-deacylglycerol reacts with inositol to form phosphatidylinositol. On the other side, CDP-deacylglycerol can react with phosphatidylglycerol, so one more molecule of phosphatidylglycerol, thus forming cardiolipin as a dimer of it. So let's summarize it. So the first step is activation of hydrophobic tail of phosphatidic acid. The activation is achieved and completed by the reaction with CTP, producing CDP-deacylglycerol. CDP-deacylglycerol reacts with inositol or glycerol 3-phosphate, thus forming phosphatidylinositol as a mature molecule, or phosphatidylglycerol phosphate, which has to be subjected to one more hydrolysis to give phosphatidylglycerol. 2 phosphatidylglycerols produce cardiolipin. And to conclude today's lecture, the biosynthesis of sphingophospholipids is based on the biosynthesis of ceramide as a sphingosine derivative, and the basis of the structure of of most sphingophospholipids. So the biosynthesis of ceramide is completed upon condensation of palmitoyl coenzyme A and serine, thus producing 3-ketosphingosine molecule. The reaction is catalyzed by 3-ketosphingosine synthase, and pyridoxal phosphate is required for this reaction. In the upcoming reaction, 3-ketosphingosine is reduced to sphingosine. Reaction is catalyzed by 3-ketosphingosine reductase, and the donor of reducing equivalents is NADPH. The next step is acylation of sphingosine with fatty acyl coenzyme A to produce N-acyl sphingosine. Reaction is catalyzed by acyl coenzyme A transferase. In the last step, N-acyl sphingosine is oxidized to ceramide by dihydroceramide reductase, and FADH coenzyme is an acceptor of reducing equivalents. So ceramide, as the basic structure of sphingolipids, is now transformed to final products. I'm gonna mention only the synthesis of sphingomyelin because we mentioned its importance, this synthesis is completed from synthesized ceramide reacting with the molecule of phosphatidylcholine, again synthesized and following the previously mentioned mechanism. The enzyme catalyzing this biosynthesis is sphingomyelin synthase. So this was the end of the story on metabolism of lipids. So now we have completed another huge chapter on metabolism, and I hope this was not difficult, this was not time-consuming topic, and until some other new topics and some other new chapters, new biomolecules and new metabolisms, stay healthy, stay safe, and bye-bye.